Yes, ma'am. Yes, ma'am.
Recording is on.
I'll do it.
I have already started recording.
Okay, okay. Yeah.
Ma'am,
it
we
is
can see only the
started.
first slide right now.
Now,
You go
The
to
first
fifth
slide
slide, ma'am.
itself, ma'am.
You
Please
click
change
the
it if.
fifth slide.
Yes, ma'am.
now you can see.
Yes, ma'am. The matrix
PAMI
slide we
matrix
can
is
see.
visible.
This you can see now.
Yes, ma'am.
Okay.
Scoring matrices.
Yeah.
PAM1 matrix.
Yeah. Okay. Sorry for
Yes,
that. So, okay. So here, as I have been discussing earlier, the arrangement of these amino acids, what are the ways the amino acids are going to be replaced, and how much is the per point accepted mutations, to what extent you can see that a particular amino acid can be replaced. So this matrix is the PAM1 mutation probability matrix. So the original amino acid, which is, you can see that the original amino acid is arranged in columns here across the top. These are the columns, that is alanine and all this. While replacement amino acid is given in the rows. You have the rows of the amino acids here and columns of the amino acids. They are arranged in the same order. But what the matrix is telling you is the percentage of changes which occur with respect to an amino acid over the course of evolution. So here, this amino acid alanine has changed is almost conserved for 98.7, but in 1.3% there is a change in the alanine residue. So that much is an accepted mutation. So if anything goes beyond that for every amino acid, you can see for glutamine it is 98.8, and glutamate it is around 98.7, 99.4. So this kind for glutamate is 98.7, glycine is 99.4. So in the previous slide I have shown you this particular amino acid is being replaced here in these different samples. So that is the percentage of these mutations which are possible or which can be tolerated over a period of evolutionary significance. So how these values are coming and all is not our purview right now. Why I just put this matrix is for you to understand that every amino acid has to have a highly conserved position, and if that amino acid changes, there is only a very small provision there for change of the amino acid over the course of evolution. But if this amino acid is going to be replaced or it is going to be mutated, then there could be some serious concerns related to the structure of the protein or the function of the protein. So when we discuss genetic diseases, like for instance, when we talk about the proteins, the most important amino acid metabolic disorders all of us know is phenylketonuria, tyrosinemia, and then homocystinuria. Like this, you can list out many maple syrup urine disease or glycogen storage disorders or lysosomal storage disorders. There are many disorders which are there, and we would also like to know there are many variants which are there for a particular molecule, for a particular protein. There are many types of mutations, synonymous mutations, non-synonymous mutations, point mutations. So how to look at these mutations and see what could be the percentage of mutations which are really important. And at the same time, if we want to see whether a particular amino acid is replaced, what are the possible changes which we can do. for a particular amino acid. If we want to replace when you are working with a protein and you want to see what could be the replacement amino acid for that, and what would happen if you replace the amino acid. So today's
Excuse me, ma'am.
yeah, I stopped sharing the screen.
Okay, okay. Right, ma'am.
Okay, so so in today's session, I will show you some of the tools where you can identify the types of mutations, or you can incorporate mutations of mutants, and also look at what would be the uh, accepted mutations. And okay, so for this, we have to again first let us go into the UniProt site. I'll just open the UniProt site and then share the screen with you. What is UniProt? Yes. What is UniProt? What did you use this for, UniProt? Yes. Anyone? Then nobody seems to respond. UniProt is a protein sequence database. So what do you get information from that? Freely accessible resource of protein sequence. Okay, so let me see what it is. What is other information you can get from UniProt? Is the screen sharing? Is the screen sharing?
No, ma'am, it is still blank
Okay.
again.
Same problem. Now, is it coming? Now you are able to see the screen?
Yes,
Yes,
ma'am.
ma'am.
Okay. So this is the UniProt site. So in the UniProt site, what is it that we can identify? For example, I am just giving a example of some enzyme like uh, phenylalanine hydroxylase. Right. So here there are some examples which we all visit. So if you take the human, there are around 32 entries, and this is the UniProt ID. So if you go to the UniProt ID, there you scroll down that particular window. In the UniProt ID, you will see so much information which is given here. If you go on moving it down, you have disease and variants. You can see on this particular left-hand side of the screen, I'm pointing out, I'm showing some links where you see diseases and variants. Why I'm showing you this is because when we want to look for the mutations, here there are some mutations which we can try to see how to what happens if this particular mutation is there in the amino acid sequence of the particular example what I have taken here. So you can see that there are so many variants which are possible. At position 19 there is replacement of H with Q and 21P with S, or at 36th position you can see the 36th position C being replaced by Y or D being replaced. When you look at this type of replacement of the amino acid, is there any similarity? Now histidine is being replaced by glutamine, proline is being replaced by serine. So proline is what kind of amino acid? Proline is a amino acid and serine is a hydroxyl group containing amino acid. Similarly, cysteine is a sulfur hydryl group containing amino acid, and tyrosine is an aromatic amino acid. These two amino acids are totally different from each other. So like this, we can make some list, or we can see that already we know this list. So from this list, you can make out what we could also assess what kind of mutations are going to useful mutations, or the serious mutations, or they are deleterious mutations, right? So you need to remember this. So we have different tools and softwares which can be used in order to predict these mutations, and then we will try to visualize the replacement and how the parameters may be altered if possible. Okay. So now I'm going, so this is, you can keep this in mind. So here, now if you want to do the mutational analysis of a particular protein, there are two tools we are going to compare. The first one is called as the polyphen, Polyphen, polyphen. Okay, so this is a prediction of the functional effects of the human SNPs, that is single nucleotide polymorphism. Okay, so now what is this polyphen-2 polymorphism? It is going to predict the impact of an amino acid substitution. Okay, when you replace an amino acid with another amino acid, what could be the impact that would have on the structure and function? And that will be using the physical and the comparative considerations. Okay, now here you have a window where you can go replace the protein sequences in the FASTA format. We can now, which you are used to replace, or which you can know already. You can know how to download the protein sequence in the FASTA format, and I have also shown you the UniProt where you can see the position of the mutation. Where is the position of the mutation? So in this tool, you have the protein or the SNP identifier where you can give the fill up that, or you can paste your sequence in the FASTA format. But in this, you have to give the position of the amino acid where you would get the replacement. And in this, there are two rows: substitution, which amino acid you would like to replace with which amino acid, right? Now that we have this particular list in the UniProt, let us only try to see these replacements of the amino acid. So first we have to get the FASTA sequence of the particular protein. So let us get FASTA sequence. So you have the go to the sequence on this top. You have the go to sequence here. Okay. You have the download button. This is the FASTA sequence. You can take the FASTA sequence of the particular protein and move to your polyphen window there, and you can paste the sequence in the FASTA format. So this sequence has come here in the FASTA format. And now what is the position where you would like to replace the amino acid? So let us go back to our UniProt where we will look at the diseases and variants section. And here I will select one particular position here. Let us see if this works out for us. At position 21, we will replace P with S. So here you can give the position of the amino acid, okay, and you have to replace the P with S. Are you following, all of you? So what I have done is I have pasted the FASTA format here, and I have taken the position of this amino acid where I would like to replace the amino acids, and which amino acid I would like to replace. I would like to replace P with S, right? And then I will be submitting the query. So it will take some time. So you have this particular result which will be coming up here. You have the job which is going on. So like that, you can try it out by using or replacing another amino acid position. So be sure that when you come out with this result, when this result comes out, we will see what could be the output of this particular result. Then we will understand what is the type of mutation this will be bringing about. So like that, you can see how many of these mutations are really deleterious mutations or not. Similarly, there is another resource which is called as the SIFT. Okay, so this is another resource or sorting intolerant from tolerant. It would be telling us whether this mutation is a tolerant mutation or it is an intolerant mutation. So this is a resource which is if anyone is parallelly working with me, you can tell me whether you are able to access this window in the chat box, or you can let me know so that we can proceed further. Yes. Okay. So I'll go to shift sequence here, select on shift sequence. Okay. So the name of you can paste your protein query sequence. Since already I have copied it, I'm just pasting the sequence here. And you can enter the substitutions of interest. So here you have been given the format how to give this substitutions here. You can give your substitution M1Y K3S T4P like that. So which means that if I'm trying to give the same type of mutation from here, what is that I should give? I should give this as P21S. I give this as I will enter the substitution P21 because we want to replace proline at position 21, so that you have to keep that in the uppercase. Okay. And then we will submit. Okay. So you will can see this link later. So let me see whether polyfen has come or it has closed. I don't know polyfen result. You can see the results for the polyfen. Okay. So, is everybody able to see this result? Is there no response from anyone? Screen is not sharing, is it?
We can see, ma'am.
Yeah.
Visible, ma'am.
Yeah. What about response from people, members? Okay, so this is the result which is coming from this polyfen 2. So when we replace at the position 21 proline with serine, this mutation is predicted to be probably damaging with a score of 1. So this is the output which we are getting for the polyfen for this replacement of the amino acid. If you are trying to replace the other position is there, so we have tried with this P2S and it is going to be deleterious. Probably it is deleterious. Let us check our SIFT result also whether we are able to get anything in the SIFT result. SIFT result is sharing. Is the screen sharing?
Screen is sharing, ma'am.
Is only
Probably
poly,
damaging
poly,
with a score of
polyfen
1000
is
C.
only sharing. Polyfen
Polyfen
result
2
is
is only
sharing.
sharing. Ah,
Polyfen 2 is only sharing. Now SIFT is sharing. SIFT results are sharing.
not yet, ma'am. Maybe net is slow, I think. Now it is visible.
SIFT results are sharing.
Ah,
Okay. So here you have what is the position we have entered for that replacement. We have seen the position is 21. Here we have changed the position for 21. So this is also predicting that this amino acid with probabilities of less than 0.05 are predicted or greater than this is 0.05. These are predicted to be deleterious. If the probabilities are less than 0.05, these are predicted to be deleterious. So let us see these positions 1 to 100 here. Now you can, is the screen visible to all of you? There is predictions for the position 1 through 100. So we have been giving this position of this 19, 21 where we are looking at the proline. So this particular position it is going to tolerate only these particular mutations which are given on this side of the tabular column. If the value is going to be as we have seen in the previous result, if this value is going to be less than that, then only the mutation is deleterious. So this threshold for intolerance, here you can see the threshold for intolerance is equal to 0.05. So if the prediction which is not tolerated is all these predictions are not tolerated, these are the predictions at that position which are tolerated. So what do you understand from this? What do you understand from this? Somebody should respond today. Yeah. Yeah. So what is your conclusion from this? Which one is predicting better, polyfen or SIF2? Hello? Am I audible?
Yes, ma'am, you're audible, ma'am.
Shall I move on to the next? How to identify the mutations using polyfen2 and SIF2? SIFT resources is what we have seen so far. Next, we will move on to the prediction of the structures. We have to look into how you can mutate the amino acids and how you can also do the energy minimization of the proteins, whether this change in the amino acid will change the energy levels of the protein. Shall we go ahead with that?
Yes, ma'am.
Yeah. Okay. So now we will move on to the all of you have installed the SPDBV in their systems. If anyone is working,
Yes, ma'am.
okay. So okay, all of you can open the SPDBV, and before that, I will. You also need to download your um, sequence. Is the screen sharing?
No, ma'am.
It's not sharing.
No, ma'am.
Hello.
Now it's sharing, ma'am.
Okay. Now it's sharing.
Yes, ma'am.
Okay. So now let's go. Let's go to the PDB database, and there, what is the protein we have just seen? Let us try to retrieve the structure of one of the proteins which we have just discussed. Okay, let me check for Homo sapiens. I will select. Are you able to see that I'm working with the PDB?
I'm not able to see till now, ma'am.
Screen is not sharing.
No, ma'am. Ante maybe you're sharing it, But net tissue, I think so, ma'am. It is very, very slow, I think. I'm not able to see. Even some of the participant has written no.
Now it's sharing.
No, ma'am.
Anything you are seeing now?
Yes, ma'am. PDB.
Mm.
One
Yeah. So
WX5.
yeah. So again, I will select Homo sapiens here, and then we will see the structures of only the human type. So we need to download this PDB format into your local computer. So this is the crystal structure of the human tyrosinase protein 1. So let us see what we can do with this. Now we can go to download files and check the PDB format here. So this will be going down into your downloads. It would be there in the downloads folder with this number, that is 5M801 PDB. So the PDB file for us is 5M80. Okay. So now we want to do, let us first look at the protein structure. So this kind of exercise you would be doing subsequently also. So for this class today, let me just share the screen. You are seeing the SPDBV viewer.
No, ma'am.
My desktop is seen.
No, ma'am.
Is it sharing now? Is it sharing?
No, ma'am.
No, ma'am.
Now it is visible, ma'am.
Now you can see.
No, ma'am.
Is the screen she seeing?
Yes, ma'am.
So, so let us, is everybody following this? So let me open this file in the downloads. So just we have downloaded it into the local computer. Now I am opening that in the Swiss PDB viewer. Okay. So you would be getting this kind of structural um details. Okay, so what this is the structure of the protein which we have just downloaded from the PDB viewer. Is a .pdb file. Now, why we are doing this? Doing this is we would like to see whether what is the energy associated with this particular molecule and what we can do to minimize the energy. So why we have to do the energy minimization of the protein structure when the protein is going to be prepared for docking with the ligand, or whenever there is any change in the structure of the amino acid which is being modified, it could lead to the change in the energy parameters of the particular protein. So in order to see that, we will find out the tools which can be used in order to minimize the structure of the minimize the energy which is related to the protein. We will be reading more later also. So now here, these are the toolbars, tools which have been explained to you yesterday by Dr. Kamala ma'am when she was introducing this Swiss PDB viewer to all of you to visualize the protein structure. So it is giving you the information which is related to the protein three-dimensional structure of the protein. So you have different tabs which are here. So you have the edit tab, tools tab. Okay, so once you have taken the structure of this particular protein, open the protein into the Swiss PDB view using the Swiss PDB viewer. Once you open the protein structure, you can also look at the control panel here, which is there in this particular tab. That is the control panel, which is this nothing but this one which is there on the extreme right side of this screen. So where it is giving the positions of the amino acids, what are the various amino acids which are there, where you can do the required positions, or you can select a particular amino acid, and you can also see the levels of the energy which is associated with that particular protein. So without doing anything, once you have recovered or once you have taken the hello, yes,
Could be.
yes,
Ma'am, please carry on, ma'am.
okay. So here we will be looking at the tools. We can go to the tools. I'm not changing anything, any parameter here. Let's go to the tools and you can find out what is the actual energy associated with this protein. So you can do different methods. One is called as the threading method and another is the force field method. So threading is actually related when you want to find out the best possible folding of the peptide and with what other proteins it is going to share its structural similarity. Then we will be looking at this threading. Now we will just looking at the force field process. We can select the compute the energy by using the force field technique. So you can we want energy of the bonds, energy of the torsions, energy of angles, everything which is there in that particular protein. So you can generate a report. It would take some time to generate the report, at the end of which you may be you will be getting the output. So by the time this is done, I will just show you the output. Again, if I Okay, so okay, so now this window has come up. In this window, you can see that these are the amino acid residues which are there. These are the numbers of the amino acid residues. These are the energies which are associated with the different bonds, the bond angles which are there, the torsion. Torsion is the amount of strain that molecule is going through because of the linkage with other side chains and electrostatic constraints. So towards the end, if you scroll down this particular page, come to the end of this page, you will be finding a total value of the energy which is associated with this molecule. So this one is around 79 minus 79,491 1.172. So this is the energy which is associated with this structure. Right now, let us remember this. Actually, I have put it into the PPT, but now if I share that, this may go. So here, what we can do is we can select particular amino acid now with
Padma
respect
ma'am,
to, yeah,
um, I didn't see that result, ma'am, because I don't know whether
result is
you've
not
shared.
result is not seen.
No.
Now is it seen?
Microsoft Teams is seen. Ah, yes, ma'am.
Output has come.
Output is there.
Yeah. So here you can see that this is the energy which is associated with this particular molecule, that is around 79491.172. So now we have this particular model. I will select a particular amino acid, that is we will go back to our toolbar. Okay, so I will go. Now I have selected a particular amino acid here. Now I will use the tools again, do the energy minimization with respect to that particular amino acid. What is how the uh, energy of this bond or energy of this protein changes once we have tried to minimize the energy. So what is the basis? What does this program do? Half an hour. Half an hour. Sorry for that. So this I have, it will again go through this window. So meanwhile, I will try to show what could happen. So this when we what would happen when the energy minimization is done. So the torsion angles or whatever the arrangement or adjustment of these bonds are there, these would be undergoing some kind of rearrangement so that the energy associated with the bond angles, all the bond energies would be minimized. So by the time that is getting done, let me see if I can share my screen. Is the screen sharing?
Yes, ma'am.
Is the screen sharing?
Yes, ma'am. Rami
Yeah.
matrix.
So yeah, here now I'm showing the output of this particular uh, minimization. You can see this is the output which is there before minimization, that is 79491. We have selected a particular amino acid there. So after the energy minimization, you are getting an energy change, a slight change is there, 79505. Now, which is lesser energy in the between these two, between 79491 and 79505. 79505 is lesser energy compared to 79491. So this molecule has undergone an energy minimization. So once the molecule has undergone energy minimization, okay, still this is going on, so let it complete. In that, when this completes, you would be getting a similar kind of output, and then you can go to the file and you can save it with a you can choose a option which is there as, unless this is completed I cannot show you. So you can go to the file option and select the current layer and save it with a different file name, that is energy minimized format. So this kind of process, this energy minimized file, you can later use it whenever you are going to do some docking or any other use where you need a energy minimization of the protein structure. Right? So once this is done, then we will come back to how you can mutate a particular amino acid by using the SPDB. Let this get done. So let us move on to the next. We will come back to this after some time. So let us keep moving further. So till now, all of you are following whatever I have shown. I have shown you what is mutational analysis. Mutational analysis we have done by using two different tools. One is the polyfen tool, where you can provide the substitution of the amino acid, and the SIFT tool, that is the tolerance, separating the tolerant from the intolerant mutation, where you can replace the amino acid and see whether that mutation is a tolerated mutation or not. What is the output? You can compare between the polyfen and the SIFT, whether they are giving the similar kind of result. You can tally with your analysis. And then we have also moved on to minimization of the protein, energy minimization, which is using the SPDB viewer, so that you can minimize the constraints which are there in the molecule. So in the background, in this energy minimization process, there will be a lot of arrangement or changes with respect to the hydrogen bonds or with respect to the van der Waals interactions. The sum of all these interactions, non-ionic interactions, ionic interactions, van der Waals interactions, all this will be the energy associated with this molecule. So in the result, we have seen that the bonds are there. So many columns we have seen the values are there. So you can observe in this slide also, there will some parameters which you can see that there is a mild change in the energy values, as I have put this result here before minimization and after minimization. So there will be a minor, very little change, as it is evident from these two numbers which are given here. So if you are choosing some other amino acid constraint or any other position, you would be able to get more changes in the energy level. So the amount of energy associated with this molecule, that should be kept minimum, so that it would be possible for you when you are doing the subsequent analysis with that structure. Okay, so now that is one tool which we have used for minimization of the protein structure. Let me see if this is done. Still it is going on. Okay, if it is done by the time I complete the other part, we will come back. Okay, now let us see the other way of using the analysis. We can go by using the Galaxy tool also. We can use galaxy.org. if you have already registered or if you have opened it, you can log into the Galaxy. And in the Galaxy, we can also do the energy minimization by using similar like what we have done other tools we have used in the Galaxy. Galaxy is also a very useful tool where you don't need to save the energy conserved form or any modifications you would like to do in the protein environment in with respect to the hydrogen ions, with respect to the salt ions, or anything in changes you would like to do in the environment of the protein. You can select that particular parameter, and this sequence would automatically be saved in Galaxy. And when you go on to the next step of the analysis, it would be taking the input file on its own, as we have seen with other tools of Galaxy. So here, first I have to like to share the steps which are involved in the determine energy minimization steps which are using for the when we are working with the Galaxy. Is the slide sharing?
Yes, ma'am.
Yes, ma'am.
Okay. So first, here also, this is the first step we have shown. I'm going to through these steps, then we will come back each step. First step is to get the PDB file, that is already you will be downloading that. We have downloaded the PDB file into the local system. So you get the PDB file into the Galaxy window. Okay. So to get into the Galaxy window, let's move to Galaxy. So you know how to get the data into Galaxy. Okay, so this is already what is done. Let us do it once again here. So get data. You can select the PDB here. Type in the search bar, search tools as PDB. It would tell you get the PDB file from the protein data bank. So now we have to give the accession code of this particular protein which we have just worked with. So what is the accession code of that protein which we have worked? It is 5M80. So make sure that you are giving the correct numbers there. Okay. So you have to get the data. So in this particular window, as I have shown in my slide, there is no changes which you need to do in any other parameter. Just you have to get from the PDB, and then you can run the tool here. Click on run the tool. So it will be running the tool. So you can see that it is working on that, and it would be getting the PDB file. Now the PDB file has come. So what is the next step you are going to do in this? The next step which you are going to do, next step what we are going to do is select the search in text files tool. So in this file, we will need to include some parameters. We have to make some modifications. You can make a note of these modifications. So here you would be having a select search and text file. So you are preparing this for the for next state level of analysis. So automatically it will take the input file from here. So the input file is the PDB file which we have uploaded, and we have to select in this window. We have to select that don't match, and the other parts you can keep it as it is. Here you have a expression, the heteroatom and match type. All this can be kept as it is. So now you can go to these tools and search text can see this particular tool is there, search in text files. When I click on search in text files, automatically it is taking this PDB file which is there. And in this particular window, next window, I told you we need to select this or shift it to don't match. Okay. And here the regular expression window, you need to change it to heteroatom. That change you need to do there. So you need to put it as HETATM. Yesterday you have been shown about all those how the panel looks in the protein structure. So it is the heteroatoms which are there, and you can see that the rest of the parameters, they are the default parameters, case insensitive and all this we are not changing there. So we can just now we can uh, go and click on run tool. So this would be taking the input file, that is your PDB file, and it is going to do your modifications whatever you have instructed it. And then the next step of this, so by the time it gets done, let us see the next setup. So here you have to select the second step is the one which we have done now. Step 2 is select the search in text files tool. Next we are going to go into this GROMAX. Now here GROMAX is one of the major tools which will be doing all this energy minimization. Even the SPDBV, if you see, that also uses for energy minimization the GROMAX versions only. So this GROMAX is going to be doing different types of preparation of your protein sample for energy minimization. So there are 3 stages which it is going to do. The first one is the initial setup on the PDB structure. Once you have taken the PDB into the search in text files, and then you have given the type of expression, all these, once they are done, this GROMAX is the tool which is going to do the initial processing of the protein structure, and then it would be configuring, and then it would be doing the solvation. Solvation is adding some ions and making it soluble, and further to that it is going to do the energy minimization. So it is going to go further down, but we will not move into all those steps because those steps are all part of the molecular docking. Once we are working still energy minimization, so I am just showing you the methods which will be there till the level of energy minimization. So GROMAX will go further also, where you can actually see the potential energy. You can also visualize how much potential energy is associated with the proteins and associated with the molecule and all that. But since it is not in your purview now, I will just go through these next 3-4 steps. First, we are looking at the initial setup, where it will take the what is the input file for the setting up of the topology and the structure. It will be the previous step of the analysis, where you have taken the text files and you have seen the heteroatoms and all that. So that would be the input for this particular step. Okay. Let's move to Galaxy and see if this step has been completed. So the search in text files is there. Now the next step which we have to go for is the GROMAX. Okay. So we have to look into the various steps which can be used for further processing of the protein structure. for minimization of the energy. So now you can select the parameters what I have shown you in the previous slide. What are the various changes you need to do? What are the tools first we have to select? And so here we will be looking at the initial setup of topology and the growth structural file. So it is going to look for the input data. The input data is the search in the text files. Then all these parameters we are not changing anything now. You can, if you need a detailed log, you can also set up the log file. So right now we are not setting up the log file. We are only taking this particular parameters, and as I have shown you in the slide, this we are not changing the SPC by E is a model for simulating the water molecules. Then this is a force field process or a force field algorithm which is not in our purview to understand right now. So just keep this default settings of the system like that, and then you will click on the run tool. So this will generate three types of files which are here: the setup file and the GRO file and the ITP file. Okay, so we can later for next structure configuration of the protein molecule. So now let us see what is the next step. Is the screen sharing?
Yes,
Screen
ma'am.
is sharing.
Yes, ma'am.
Galaxy
Next,
is
we are going to the step 4. This is the step 4 where we need to use the GROMAX structure configuration tool. So this structure configuration tool, you have, this is the next one. We need the initial setup. You would be having this structure configuration tool. So this format which it is going to give you is the input file is from the previous one. So here you need to change the dimensions, the box dimensions, which is generating the solvation and other things subsequently configuration of the protein structure. So you can need to change to 1, and the box type you have here as rectangle box, and here uh, on this to generate a detailed log. So this step 4, you have to do some modifications in the tools, which in the tool you need to give some instructions to be related to the configuration of the structure. Let us go back and see if this particular window it is still going on. So this is the structure configuration file. So you can see that it would take this particular GROMAX setup. What is here? You can see this particular data here. So this is the setup. In this window, you are not visualizing anything. It will be only giving you the parameters which are there. Okay, so let us now go to the next aspect which is there. That is the GROMAX structure configuration. So in this GROMAX, you are taking this particular file is taken as out of the 3 files which are there. It would be taking only the GRO file as the input for this next step of structure configuration. So output format is the GRO file, and in this I have shown you the configure box you have to select, box dimensions and box type. So here, so configure box, you need to switch it to S. Box dimensions, you need to save it. You need to save the box dimensions as we have given it here to 1. Change the box dimensions to 1. Okay, box type, and also this detailed log which is switched off, this you need to put it on, say yes, and then you click on the run tool. So this again is going to modify the configure the protein structures in order to minimize the energy. So this will be working at the back end, and these are all some mathematical equations for calculations which will be done based on the bond angles and so on. So the next step which we have to do is the solvation. Okay, so here this has been completed. So the next step which we need to do is the solvation step. Okay, so solvation step, when we are doing, we have to look at different parameters which are necessary here. So this one is going to select the topology file. Previously, it has taken a different input. This that is advantage of using the Galaxy. The tool which you choose, whatever the tool you would like to choose from the toolbox, this Galaxy will be taking the appropriate input from the files which it is generating. So even if you are not sure which file to take, it would be prompting that what is the data on which it is going to do. In case you would like to change this particular parameter on which you want to work, you can select a different file. What You can select a different file by browsing in this window. That is, we have to go for the solvation and adding ions. So in case you want to select a different one, you can choose any of these files. So that is a way we are going to do here. So now, what is the input file for the step 5, which is the solvation tool? That is the top file. So we need to select the top file from this data. So we will go back to this structure configuration GROMACS solvation, and this GROMACS solvation will be taking up the TOP file, and then we need to do some modifications in the rest of the parameters, whether you want to add ions to neutralize the system, what is the water model, which this is based on the different configurations which are there in the tool, and specific salt concentration you want to add, and how much. All these are the parameters. We are not specifying any salt concentration. In case you want to change the solvation, or you want to change any other parameter, how much water is going to take up, or how much the ion concentration, you have a specific ion concentration at which you would like to work, then you can specify the salt concentration. So here we are taking these values by default. Okay, so here this log, we have to click it on yes, and then we would be running the tool. So the next step is the energy minimization step.
It's
So this is the 5th step in which we are doing. This is the last step, energy minimization tool. So in this, the input file is the structure file, the topology file. Then here we are not changing any of these parameters. What is important here is all these are the electrostatics parameter, distance for the cutoff. All these are basically mathematical steps which are required. Short range van der Waals cutoff. What is important for this is how you are energy minimizing, how many steps you are running this particular structure to minimize the energy. So this will prepare the protein for the subsequent simulation studies or other higher end studies. So the number of steps it is going to train the model or train the protein structure in order to minimize the energy. So that is the steps which we have to select. Here we are selecting around 50,000 steps. It is going to repeat the cycle and then it would be putting out the output for us. So 1000 is the tolerance limit, then the minimum maximum step size. All this will be the default values. What we can change is the number of steps in this energy minimization step. Okay, so you can see, visualize any of these data if you are in wanting to see what is there, what could be the result of this. It is you can visualize it also here. You can see this is the gram solvated, and you can see what are all the parameters it has worked. It is generated so many. The 330,000 non-bonded parameter combinations. It has developed three bonded neighbor chains, protein chain A, excluding two bonded neighbors. So it is doing all these back end work for our molecule, which can be visualized. But that is why we would be saying the log file, which will be telling us what are the parameters it will be doing. It can it is going to tell you the steps which it is performing, replacing 44 solute molecules in the topology file. So if you are working with these files, then you can look at this particular log file and you can do the parameter, change the parameters according to your requirement. So now move on to this next one. So the GROMACS energy minimization. So here, this is the parameter input. Let us see, go back to our file here. So here it is parameter input is the one which you have to change. Use default partially customizable setting. So you can change that in this particular window. You can change the parameter output, use default partially customizable setting. Okay, and then yes, is it not sharing?
కాలేజ్ అడిగి sharing, ma'am.
Happiness is not sharing.
మాట్లాడుతుంటే టైం లేకుండే ఆమె వేరే వర్క్ అట్లా ఇంటికి
Hello. Okay.
ma'am, carry on, ma'am.
Yeah. So you have this choice of integrator, steepest descent algorithm, all these parameters, you can keep it as it is. As I told you, you can come here, number of steps which you would like to do. This one, you can put it at 50,000 steps. This is editable. You can edit here. And same way, the EM tolerance also parameter, I told you, EM tolerance parameter also you can change it to 1000. So you can change those parameters here. Okay, and then maximum warnings, generate detailed log, and then you can run the tool. So this will be the final step for the energy minimization process. In the case that we are working, you can see what all GROMACS can do on this side. You can see we are stopping at this particular step, and you can continue further in other aspects of it when you are really looking at the molecular dynamics, that when you are trying to see the interference docking, or when you are taking a ligand and putting it with the protein, and then you would like to see what is the further status of that. Since we are not right now, you are not in the you have not yet gone the background of all that, and it is not purview here. I'm just showing this that this is one of the tools which you can use for the energy minimization also. Okay, so let it go on. So meanwhile, let me see whether I have got the output. So this is the output which we have got after the steps of energy minimization, which we were doing after selecting the particular thing. So you have a change in the energy levels. If you have saved the previous one, then you can compare this with the previous one and see what could be the changes in the amino acid structure, the protein energy levels. Okay. I'm not sure how much all of you could follow. If you have not followed, you can ask me any step. Was it very fast or it was little difficult this time? I know because the um, internet issue, I could